Hello, my dear students. Welcome to my today's online class, that is continuation of recombinant DNA technology topics, paper 6. After having learnt the various aspects of recombinant DNA technology, now it is the time for us to learn the applications of RDT or applications of biotechnology as per your syllabus. One such application happens to be transgenesis. Now in this class we are going to study about transgenesis and its significance, knockout gene mouse production and knock-in mouse productions. Okay, how do you produce transgenic mice? So this is the quota for us. Let us see how much we are going to complete today. So this is first slide with my introduction. I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bangalore 1. You can see my cell phone number and email ID. You know why. After the talk, any queries, you are most welcome to contact me through any of the avenues. Okay. Now let us start the topic as such. What is transgenesis? Transgenesis, by definition, refers to the phenomenon of introduction of exogenous DNA into the genome to create and maintain a stable heritable character. So the definition says you are going to introduce the gene of your choice, that is exogenous DNA into the genome of the animal which you want to make it transgenic, so that once the experiment is over and you get a transgenic animal incorporating the gene of your choice, you are going to create and maintain a stable heritable character in that transgenic animal. That means once it starts reproducing, it will pass on the character of the DNA which you have inserted to the next coming generations. That's what the definition says. So I repeat, transgenesis refers to the phenomenon of introduction of exogenous DNA into the genome to create and maintain a stable heritable character. And you should know certain terms right now. The foreign DNA, what you are going to introduce, is called transgene. Okay, transgene. Then the animal whose genome is altered by adding one or more such transgenes is said to be transgenic. Say you have mouse with you. You are going to add the exogenous DNA through RDT experiments, and once the experimental animal gets this gene imbibed into its genome, then the transgenic mouse what you have produced is no more the wild type, but it has acquired one special character or two special characters depending upon how many transgenes you have introduced, and now the animal is said to be transgenic. We don't say wild mouse, we say transgenic mouse. Okay. Then the transgenes, once they are into the genome of the transgenic animal, they behave just like any other regular genes of that animal, and during reproduction they are passed on to the offsprings. That's what I meant by you are going to incorporate a stable heritable character in the transgenic animal. Okay. Then thus transgenic animals are genetically engineered or genetically modified organisms, GMOs. What is the speciality? They have a new heritable characters incorporated into them. How the new character came? By the gene what you have introduced. Okay. So this is what happens during the process of transgenesis. What is the significance? A favorite question of paper setters. Often they ask you to list the significance of transgenesis either for 3 marks or for 5 marks. It's a powerful tool for studying gene expression and developmental process in higher animals. It serves for the improvement of genetic characters of animals. It serves as good models for understanding the human diseases. Okay. Then several proteins of interest which have been produced by transgenic animals are of medical and pharmaceutical significance. So they have lot of medicinal importance, right? Then transgenic farm animals, not FARM, please note, PHARM animals. Transgenic farm animals today happen to be a part of lucrative worldwide biotechnology industry with great benefits to mankind. Okay. Then transgenic animals are important for improving the quality and quantity of milk, meat, eggs and wool production that normally the human beings use, which are of either food value or used for other purposes. It helps in creating drug-resistant animals. So these are the significance of transgenic animals. After having understood the definition of transgenesis, after having learned few new terminologies, after having learned the significance of transgenesis, now we should learn transgenesis in mice. How are you going to produce transgenic mouse? Okay. Now, even today, mouse continues to be an animal of choice for most of the transgenic experiments. Why? Why means they are small, easy to handle and researcher friendly. Okay, it is easy to maintain the mice colony rather than an elephant in the laboratory, right? Therefore, they are researcher friendly. And they produce more ova. Okay, they produce more ova normally in one estrous cycle, that is the reproductive cycle, which the non-primate females practice rhythmically, that you will learn more about it in developmental biology class. So in one estrous cycle, a normal mouse produces 5 to 10 ova. That means potentially capable of giving birth to 5 to 10 pups, provided there is successful fertilization. Right. On the other hand, if you make them superovulate, okay, it is possible. I will tell you how they can be made to undergo superovulation. If you make them to superovulate, they can produce as many as 40 ova. Okay. This is the reason why, again, they happen to be the animal of choice. Thus, the methodology of transgenesis is well developed and standardized for laboratory mice and rodents. Okay. After knowing why the mice happen to be the animal of choice for our experiments, now you should know how are you going to produce transgenic mouse. Okay. You can produce by 2 methods, either by adding extra gene, then also it becomes transgenic, or by removing its regular gene from its genome, then also it becomes transgenic. If you introduce the exogenous DNA or foreign gene, then the technology is called knock-in technology. Okay, introduction of the genes is called knock-in technology. In contrary to the knockout technology, where you remove one or two particular genes to know their particular functions. Okay. First we shall study in this class how are you going to produce transgenic mice by knock-in technology. There are three methods for introducing a foreign gene into the mouse, and this is called knock-in technology. First method is retroviral method. Here you will make use of retroviral vectors. Retroviruses are a group of viruses which have been exploited to design vectors for RDT experiments. So these retroviral vectors can be made use of to produce transgenic mice by knock-in technology. So in this method, normally transfer of small pieces of DNA around 8 kb can be effectively carried out. But if your aim is to transfer gene of bigger size, this method is not useful. You have to go for other methods. I told you there are three methods. You have to go for other methods. This method is useless. So it is unsuitable for the transfer of larger genes. Okay. Up to 8 kb it's okay. Even for small genes there is a risk. There is a risk of losing some regulatory sequences which are very important for other processes, right? So there is a risk of losing some regulatory sequences. Then, apart from these two, there is risk of retroviral contamination, especially if you are aiming to produce food products which are often used for human consumption. Because of these limitations, this method of production of transgenic mice through retroviral vector method is not in regular use. Okay. Then the second method happens to be the micro-injection method. In my previous class, while discussing about physical methods of DNA transfer, I have taught you three methods: micro-injection, liposuction and electroporation. I hope students by now you are familiar with the micro-injection concept. Okay. Let's see what exactly is done in micro-injection method. The young virgin female mice. Young means virgin means the females which have not copulated with the males. They are called virgin females of 4 to 5 weeks age, okay, around 1 month, right? So the young virgin female mice are made to superovulate. What is this superovulation? Normally, in one estrous cycle, I told you they produce 4 to 5 ova, but you can always make them to superovulate in the sense you can give the hormones from outside source. This can be achieved by exogenous administration of hormones. You must be knowing a hormone called FSH, that is follicle stimulating hormone. You must be knowing another hormone called PMS, pregnant mare serum hormone. Okay, mare is a female horse. Okay, pregnant male serum, right? So you can administer, you can give the injections of either FSH or PMS to the mice, and this has to be followed by exogenous administration of another hormone called HCG, human chorionic gonadotropin, after 2 days of administration of either FSH or PMS. So what you will do? You will take young virgin female mice. You will give the injections of either FSH or PMS. All dosages need to be fixed, maintained very thoroughly. Okay. And after 2 days, for these mice which have been already administered with FSH or PMS, you will give additional dose of HCG, that is human chorionic gonadotropin, after 2 days, so that the mice which have been treated with these hormones are ready to superovulate. That means instead of releasing 4 to 5 ova from their ovaries, they will release 40 ova. This is called superovulation. Okay. The superovulated mouse produces 30 to 35 eggs or even 40 eggs. Okay. So on one side you have the superovulated mouse. Okay. So which will release 30 to 35 eggs. Okay. Then, so figure 1 will depict the protocol of production of transgenic mice through DNA microinjection. What exactly is done? The superovulated mice are mated with males. Okay. The superovulated mice are mated with males, and next day, of course, in the estrus phase of the estrus cycle, otherwise it's a big waste. And then next day, these mice are sacrificed. That means killed. Sacrificed means killing. They are sacrificed under laboratory conditions, and after sacrificing, you can retrieve the fertilized eggs from their fallopian tubes, or what you say, oviducts. Okay. So first you will make the mice to superovulate, then you will make them mate with the males, and next day you will sacrifice them. Then you will cut open the fallopian tubes and you will retrieve the fertilized eggs. So you can see the fertilized egg here in the diagram 1 in the third step. Now for this fertilized egg, you will use microinjection technique, okay, where you will have a microinjection needle and a holding pipette. The DNA of your choice is introduced into the male pronucleus. Students, please note, in the fertilized egg you can see the 2 nuclei. The smaller one is the female pronucleus and the bigger one is the male pronucleus. You are going to inject the DNA of your choice into the bigger nucleus, that is the male pronucleus of the fertilized egg. But adequate care must be taken to ensure that when you are inserting the DNA of your choice, you have to puncture the elastic nuclear membrane, but at the same time you are not going to damage the nucleoli. Okay, you are not going to touch the nucleoli. The needle should not touch the nucleoli. For this purpose, if you want, a dissection microscope can be used for identifying the male pronucleus and for microinjection. Then the eggs with transgenes are kept overnight in an incubator. You can't directly put it into the other mouse or the faster mice. The eggs with transgenes are kept overnight in the incubator, okay, so that they develop into a 2-celled stage by the process of cleavage. These eggs in 2-celled stage are implanted into a foster mother, ಬಾಡಿಗೆ ತಾಯಿ ಅಂತೀವಲ್ಲ ಅದು foster mother, that is pseudopregnant female mouse. How are you going to produce them? A pseudopregnant female mouse or a foster mouse is the one which has been mated the previous night with the vasectomized or infertile male. Okay. The foster mother can deliver pups after 3 weeks of implantation. You know that gestation or pregnancy period in rats and mouse is 3 weeks. Okay. So please understand what you did. You will take the fertilized egg. You will identify male pronucleus and female pronucleus, and through microinjection technique, you will insert the DNA of your choice into the bigger nucleus, that is the male pronucleus. Then that you will incubate so that you will get 2-celled stage embryo. Now, on the other side, you should keep foster mothers ready. How are you going to do it? See, normally all rats, cannot implant the fertilized eggs artificially. They need to be kept ready. Okay, their uterus need to be primed up for the acceptance of the fertilized eggs. For that, what you have to do, they should get ready. That means they should be mated with the male ones, but the male ones should have been vasectomized. Vasectomy is surgical removal of vasa differentia. That is a sort of family planning operation, what you say in humans. So the males should be vasectomized. They must be infertile. They should not be able to fertilize the eggs, though they copulate with the females. The moment the females are made to mate with the vasectomized or infertile male, their uterus get primed up. You know that in laboratory rats and mouse, you find bicornuate uterus to accommodate the pups, right? So, such females are called foster mothers or pseudo-pregnant females. They assume that they have undergone successful copulation and mating. Therefore, they can carry the implanted embryos. So, it's a psychological status. So, such mouse are called foster mother or pseudo-pregnant female. And once you keep these pseudo-pregnant female mouse ready, you can always implant the 2-celled stage embryos into these foster mothers. So many you have to have. Then, one of these foster mothers, there are chances that they will carry the embryo which has taken the DNA of your choice. That means, after 3 weeks, you will get the pups out of these foster mothers. The presence of transgene in the pups. Right now, we will not be knowing in which of the pups you will have the gene of your choice. So, the presence of transgene in the pups can be identified by PCR technique, that is polymerase chain reaction technique you have in your syllabus. I am going to teach you little later. Or there is one more nucleic acid hybridization technique called southern blot hybridization, either through PCR or southern blot hybridization. You can know in which particular pups the transgenes have been incorporated. The transgenes are present. Okay. And then the mouse. So in the diagram, you can see one pup with black color, just to show that that is the transgenic founder. So the mouse carrying the foreign gene is the transgenic founder, this black one, okay, from which you can establish pure transgenic lines. Okay, this is how you are going to produce transgenic mouse by DNA microinjection method. Okay, another method I will teach you little later. Now, this method has its own limitations. So, here again I have described the procedure, that is superovulated males are mated with superovulated females, sorry for the spelling mistake, superovulated females are mated with males and sacrificed on the following day. The fertilized eggs are removed from the fallopian tube. Same thing I have taught you. Then my microinjection, you will incorporate the DNA of your choice. You will take enough care not to puncture the nucleoli, right? Then, so the eggs with transgenes are incubated. Then two-celled eggs are implanted into a foster mother, right? Then they will deliver the pups after 3 weeks of implantation. Then presence of transgene can be identified by PCR or southern blot hybridization. You can establish a transgenic founder from which you can establish transgenic lines. This is what I have explained right now. What are the limitations? First one, 3 to 5% of the inoculated eggs develop into transgenic animals. 3 to 5% of the inoculated eggs develop into transgenic animals. In the sense, if you have 100 animals in which you have incorporated the gene of your choice, you will get 3 or 5 transgenic mice. This is the rate. Okay. Then random integration of foreign gene into the host genome. That means the gene of your choice may not go and sit in the place where you want it to sit. Okay. So it may go anywhere and simply get incorporated into the genome. It has its own drawbacks. Then these transgenes, since they go and sit in odd positions, they may not be expressed, they may be underexpressed, or they may be overexpressed. Okay. Then the process is time consuming, costly, and labor intensive. Okay. So these are the drawbacks of this method of production of transgenic mice. But despite of all these limitations, this technique is routinely used for producing transgenic animals. So till now I have taught you the definition of transgenesis, few terminologies what you are supposed to know, and then significance of transgenic animals, then transgenic mice, how to produce. Out of 3 methods, I have taught you 2 methods. Dear students, I am going to stop at this stage. In the continuation lecture, I am going to teach you the other method and knockout technology. Thank you for your patient hearing. Stay blessed. Thanks. My dear students, welcome to my today's online class on applications of biotechnology. In that, continuation of the chapter on transgenesis. In the last class, I had discussed about the definition of transgenesis, significance of transgenic animals, and production of transgenic mice through knock-in technology, that is adding of exogenous DNA. I had told you that there are 3 methods, and out of 3 we had discussed 2 methods. One is retroviral vector method and the other one is microinjection method. And I had told you that despite all the limitations, the microinjection method is routinely used for producing the transgenic mice. Now, as a continuation, let us learn about the 3rd method of producing transgenic mice, that is embryonic stem cell method. What exactly is done in this method? Let us see. See here, cells from inner cell mass of a blastocyst stage of a developing mouse embryo is taken and is made to proliferate in cell culture. What you have to do is, you have to take the blastocyst stage of a developing mouse embryo. In that you have to identify the inner cell mass and you have to take them out and they need to be cultured. They proliferate in the culture. These cells are called pluripotent embryonic stem cells, ES cells. Pluripotent means they are capable of differentiating into any other types of cells, including germlines, okay, when transferred to another blastocyst embryo, right? So what you will do, you will take the blastocyst of mouse embryo, collect the inner cell mass and proliferate them in the cell culture, and now you call them as pluripotent embryonic stem cells. These are capable of differentiating into any other type of cells including germline cells when transferred to another blastocyst embryo. Now, what to do with this? The foreign gene, that is gene of your choice, is introduced into this ES cell or embryonic stem cell, which in culture can be subjected to genetic manipulations without changing their potentiality, that is, without changing their pluripotency nature. Now, the foreign gene is introduced into these ES cells either by electroporation or microinjection, both of which I have taught you in my previous lectures. Okay? Then, once the desired genetically engineered cells with transgene are ready, they can be identified by selection procedures. Either you will make use of marker gene or PCR analysis, that is polymerase chain reaction analysis, or you can make use of marker genes. Both I have taught already. Then, these transfected cells can be cultured and they can be introduced into another blastocyst by microinjection. Then, these blastocysts are implanted into foster mother, that is pseudopregnant mouse. How do you generate the pseudopregnant mice? I have taught you in my previous lecture. Then, thus the transgenic founder mouse can be produced and the transgenic lines can be established by suitable breeding strategies of the founder mice. This is how you are going to generate the transgenic mice from embryonic stem cell method. Now, let me show you the schematic representation of how exactly the procedure is done. Bear with me this is what I wanted to show you. You concentrate on the figure 2, ES cell transfer. So here you can see the embryonic stem cell culture. You can insert the gene of your choice into this. Then you can subject them to clonal selection. You can take the transfected cells and put it into the female mouse, right? And then, or you can co-culture both of them and then you can put them into the recipient foster mice. And the black ones what you see here are the transgenic founders. These are the ones which have taken the gene of your choice and you can establish transgenic mice line. Okay. So this diagram will show you the embryonic ES cell culture method. Okay. Production of transgenic mice by embryonic stem cell method. Okay. Now, let us come back to our bear with me for a few minutes. Let me come back to the original slide. Yeah. So I have explained the process of production of the transgenic mice by embryonic stem cell method. Right? Now, this is about knock-in technology. Now, there is one more technology called knockout technology. Okay? Gene knockout process. There is a minor mistake in the heading. It is not knock-in technology. It is knockout technology. Sorry for this heading. Knockout technology or gene knockout. So in this gene knockout process, an existing function can be blocked by destroying a specific gene. Okay. So the target gene disruption can be carried out by incorporating a DNA sequence, namely a selectable marker gene SMG into the coding region. What exactly done in this here is the chromosome carrying the target gene with exons. By now we must be knowing exons are introns. So with exons and with flanking sequences, this is subjected to homologous recombination with a vector carrying an SMG or selectable marker gene. The homologous recombination results in gene knockout. That is, in the place of the exons, the selectable marker gene goes and sits as shown in the diagram, right? So as a result, there is disruption of the target gene. So in this technique, the loss of function occurs. Okay, loss of function occurs in contrast to the gain of function that takes place by introducing a foreign gene in knock-in technology. Okay. So this is what exactly is done in knockout technology. The homologous recombination results in knockout, that is disruption of the target gene. There is loss of function in contrast to the gain of function in the transgenic mouse generated by knock-in technology. Okay. What are the applications of knockout mice? For understanding the development and physiological consequences in an organism, these knockout transgenic mice find a big application. The moment you remove the gene, okay, from the genome, that particular function will be missing. That's how you can understand which gene is responsible for which particular physiological function or development. Then it helps us to understand the biochemical and pathological basis of several human diseases. Students, to date, around 200 knockout mice have been created to serve as animal models for the study of a large number of human abnormalities. For example, SCID mouse, that is transgenic mouse with severe combined immunodeficiency, then knockout mouse for allergy, knockout mouse for transplantation, knockout mouse with memory loss, knockout mouse with retinitis pigmentosa. Here I have shown you the photograph of the knockout mice, okay, which can be used for particular experiments. So this is all about the production of transgenic mice through embryonic stem cell culture method and then knockout technology and its significance. Dear students, thank you for your patient listening. Any questions you can contact me. Thank you. Stay blessed.